with Steve Paikin is brought to you by the Chartered Professional Accountants of Ontario. CPA Ontario is a regulator, an educator, a thought leader, and an advocate. We protect the public. We advance our profession. We guide our CPAs. We are CPA Ontario. And by viewers like you. Thank you.
It's been called biology's moonshot. 20 years ago, the first draft of the human genome was published, with enormous hopes for what could come next. Tonight, we'll assess whether the legacy of that achievement has lived up to expectations. Then, author and futurist Jamie Metzl is with us to signpost the frontier of genetic engineering and humanity's future. It's Monday, March the 1st, and that's next on the agenda. In February 2001, after a remarkable global effort, the Human Genome Project published its first draft in an effort to map the 3 billion base pairs of our entire genome. That was heralded as a massive accomplishment that would yield incredible dividends. 2 decades later, how should we understand the legacy of the project? And did it deliver the anticipated results? With us to reflect on that in Santa Cruz, California, David Hausler, Scientific Director of the Genomics Institute at the University of California at Santa Cruz, who led the team that assembled the first human genome sequence. In Princeton, New Jersey, Shirley Tillman, Emeritus Professor of Molecular Biology and Public Affairs and a former president of Princeton University. And in New York, Aravinda Chakravarti, Director of the Center for Human Genetics and Genomics and Professor of Neuroscience and Physiology at New York University's School of Medicine. And we're grateful to the three of you for joining us on TVO tonight. I do want, well, let's start with a little nostalgia. Shirley, when you go back to 1990, when this whole thing officially got underway, during the 90s, how would you characterize the kind of excitement that you and the people who were in this field were feeling about its potential?
Well, I think those of us who were involved with the project were extremely excited about the potential for the sequence to tell us a great deal about human physiology and disease. But there was a significant fraction of the scientific community who were very skeptical about the project in the beginning, who were worried that it would soak up money that would be otherwise going to other areas of biomedical research. And there were even a group who were very skeptical that we would ever understand the information in the genome. So we were gathering a lot of data that would essentially go unused.
Aravinda, how about you? What was your sense of excitement or expectation at the time?
You know, in thinking back, it's not only the scientific legacy, it actually is an emotional legacy. You have to remember 30 years ago, we were all 30 years younger. Enthusiasm came by easily, but this was even beyond any of our wildest dreams. If, in fact, it could be delivered, it would change the nature of the science I was learning, which was human genetics, which was really a fledgling field then, would change forever. And the way I describe it is, for me and many others, it opened doors where there weren't any doors. It was a new science. It was a brand new community. It was sort of like an immigrant community like New York, where it brought in engineers, physicists, chemists, mathematicians, molecular biologists, biochemists of all kind. And to me, that was one of the most exciting things that was happening.
David, some people called it biology's moonshot. What did it mean to you?
Oh, it was actually something that I was aspiring to, but as a computer scientist, it wasn't even clear how I would be involved in it. I longed to be a part of it for many years, and I joined a little bit late in the game when the going got rough in terms of the competition between the two projects, the International Human Genome Project and the Celera Project, and the need to assemble the data very, very fast. So our team was a team that came in at the last minute and helped out. To be honest, in 1990, I didn't even expect to be part of the project.
And things kind of changed after that, didn't they, for you?
Things happened.
Things happened. That's a good way to put it.
Yeah.
Shirley, I want to just pick up on what you said in the midst of your first answer, which was that, this was not a slam dunk for a lot of people. There was skepticism, there was concern. Can you just go into that in a bit more detail? What were people worried about at the time?
Well, as I said, I think the thing that they were mainly worried about is how it was going to negatively affect their ability to raise funds for their own research. But the tide really turned when the project made what I think was one of its most critical decisions, which is to learn how to sequence, because at the time we didn't know how to sequence well enough to sequence 3 billion bases of the human genome, but to learn by sequencing the genomes of simpler organisms with much, much smaller genomes, beginning with simple bacteria, going on to yeast, onto small soil worms, ultimately the fruit fly. And eventually, of course, we were ready by that point to sequence the human genome beginning at about 1998. But what that decision did was bring into the project all of the skeptics, because no matter what organism you worked on, suddenly you had genome data that you could have only dreamed about having before. And that really ended any of the resistance and the skepticism about the project.
And Aravinda, it was during that decade that computing power and computing potential just exploded exponentially. How important was that in terms of being able to achieve what you all achieved?
I think there were two kinds of achievements. I came from a mathematics and statistics background, and I often worried whether I'd learn enough biology to be able to become this new kind of scientist. But there were already many, many developments in computing, and not only that, in the science of computing. And it so happens that genetics and sequencing the human genome in particular, that this was a very information-rich science, right? Genetics is about the transfer of information from one generation to the next. And so there was a natural marriage, if you will, of developments on each side. And in a way, I think we've always described it as biologists requiring the math, stat, and computational side. But I think actually, if you go and look at the other side, there was a lot of developments in the Genome Project that helped. Some developments, applied developments in mathematics, statistics, and in computing as well.
David, I'm going to say a date, and I want you to tell us what it means to you. July 7, 2000. Go.
That was definitely the highlight of my life. We were racing like crazy to get the genome done and out there. And at that moment, I had to look back and say, here is a long sequence of A's, C's, T's, and G's. As a computer scientist, as Aravinda was saying, it's just information. But in fact, it represents the struggles and the triumphs of our ancestors since the dawn of time. I mean, this is the message that has been passed on like the Olympic torch from parent to offspring for so many millennia. And now it comes to us, our script that was forged by our ancestors. And at that moment, I realized that we had put it together. The 20 different centers from around the world have been cranking out sequence like crazy as fast as they could in anticipation of the announcement at the White House on June 26th that the first draft was complete. But that draft had not actually been shared with anybody. And because we did the computational part of assembling all of the pieces in this mad rush to get it ready for the White House ceremony, we had the honor of posting it on the internet. And again, like Aravinda said, there are two great technologies of the late 20th century, which are molecular biology and bioengineering and computer science. And they came together in that moment. We posted that first draft on the internet, and now our genome was copied at the speed of light. spread around all of the planet in a very non-biological way.
Let me just jump in with a quick follow-up there.
Heritage.
Why'd you put it up on the internet?
Oh, this is a matter of principle, really. Our competition was making it available only by subscription and claiming intellectual property on it, and we felt that that was inappropriate for that information that is so fundamental to humans. So we put it on the internet free without any restrictions on use and no claims of intellectual property.
And Shirley, when the news eventually got out, for you, what were some of the biggest surprises in terms of discovery?
Well, I think the biggest surprise was how few genes we actually have as human beings. I had been teaching for years that the human genome probably had something on the order of 100,000 genes. But when we were able to look at David's ensemble and begin to count up the number of genes, it was shocking that the number is probably in the order of about 22,000, not 100,000. And this was a humbling moment because, after all, we knew how many genes there were in these other organisms that we'd sequenced, and it wasn't that many fewer genes. So suddenly we had to contend with this huge, I think, philosophical dilemma, which is, how do you make something as complicated as a human being with so little apparent information?
Yeah, Aravind, I wonder if you could follow up on that, in as much as I guess we discovered that we don't have very many more genes than a worm. What should we infer from that?
Well, I think that says that we have looked at the biological world in a very different and biased way. It says nothing about our genome. It says something about what we believe. An interesting thing is at a meeting in Cold Spring Harbor, these were annual meetings, and on the Genome Project the year before the genome was fully assembled, there was there was a competition, if you will. There was a poll to see who could estimate the correct number of human genes. And just as Shirley mentioned, the estimates varied all over the place, but most of them were at the high end. I think the fact that we have 20,000 odd genes says that we need to figure out exactly how a human being can be made and is made, not can be made, is made from 20,000 genes. What is it about the rest of the genome that makes those 20,000 genes do all of the varied biology and physiology that we study? And so the problem shifted not to merely counting genes, but to figuring out how they created this fascinating and complicated human biology, and similarly, the biology of other organisms.
Well, that takes us to the next part of our discussion. And to set up that part of our conversation, let me just read a quote here from Richard A. Gibbs, who worked on the Human Genome Project. He had this to say this past summer on the expectations people had of the project. The predictions included the possibility to breed so-called super babies based on this new knowledge, and at the same time, perhaps even predict criminality. In hindsight, there was plenty of hype that was shared with the media and the wider community. Critics are correct that the apex of these claims was not reached. The hyperbole that we look back on did not, however, come from the front line. It came from those who championed the program, mindful of its long-term benefits. Thanks to them, they generated the enthusiasm to fund this transformative work. David, maybe you could pick up the story from there. 20 years later, has the project delivered on the hope and the optimism that originally surrounded it?
The project has opened our eyes to the genetic structure of human beings. That is the fundamental thing. There's always a little extra hype around a project of this magnitude and importance. And so I really think you can't hold that against the project itself. There are expectations that medicine and biology will be easy. One always thinks, well, the scientists can surely figure that out, but some problems are very deep. They run extremely deep. And the complexity that we're dealing with is really astounding. So it's certainly no surprise to me that medicine was not solved by sequencing the genome.
Well, Shirley, I'll follow up with you on that, because the hope was, of course, that we would now understand the genetic base for many diseases, and a gene for this, a gene for that, and potentially be able to conquer many of these things. How much of that, in fact, has happened?
So I think that there was a certain degree of disappointment when we began to use the genome to begin to understand not simple genetic diseases, but very complex diseases like heart disease, like stroke, like cancer. And the expectation was that the genes responsible for these would fall out very quickly through analyses of genomes. What we now know today is most of the really common disease that causes the vast majority of mortality in the world are the consequence of the action of literally thousands of genes interacting with one another in very complex ways and in different ways in different individuals. So one of the things we've learned is turning that genetic information into treatment and prevention of disease is going to be much harder than we thought it was going to be.
In which case, Aravinda, is that not actually a hugely significant discovery, in as much as we are apparently even more complex than we might have initially thought?
Well, there's no doubt about this. But let me speak to the success before I tell you of what happened or what's happening now in our understanding of the much more complicated disorders, as Shirley alluded to. Today, we know the genes for about 5,000 rare disorders. And for many of these rare disorders, a child who's now diagnosed can literally walk into a clinic and get a diagnosis. And this prevents, I would say, by sequencing, years and years of what is often called a diagnostic odyssey, where patients go from one clinic to the other, not really knowing what's happening to their child. Therapies are coming much slower, but therapies are happening. I think the timeline for therapies is somewhat different because the science is different. It doesn't depend only on the sequence. But the second thing to remember, beyond our understanding of complex diseases, which we now do, heart disease probably results from the effects of several hundred is thought to be contributed by 1,000 genes. But one has to remember, even as we slowly unfurl the causes of this disease, that medicine by its very nature is a conservative discipline. Sequencing is not sufficient. We've got to teach the teachers. We've got to explain to physicians. My internist that I go to as to why this information is important, how she or he is going to use it, and how therapies can be adapted to this. So the change in medicine, I think the scientific change has happened and is happening. I think the cultural change will take quite a bit longer.
David, what would be the main things on your list of things that we now, 20 years later, have a much better understanding of, thanks to the work that was done 20 years ago?
I think we understand a lot more about evolution. There were a number of surprises in the genome. Only about 5% of it represent nucleic acids, the A's, C's, T's, and G's that are actually under selection by natural selection. The other process that's going on is that there's essentially selfish DNA, these DNA pieces that copy themselves for their own sake. It's almost like a parasite on our genome. And there's been a battle that has been going on for eons between our own genes and this other type of DNA. And we are kind of forged from that battle. It's created a system that is quite robust. There are regions of the genome that have been highly conserved. And one of the surprises was these weren't even the genes. They were the regulatory elements that Aravinda was referring to. These parts of the genome create this complex interaction that makes us us. And it is quite subtle and quite different from the way you would have imagined designing a phenomenon like this. It's not engineered by us. There's a very alien engineer whose touch is evident when you try to understand the genome.
I got to follow up on that. What does that mean?
That alien engineer is natural selection.
There we go. Shirley, how about for you? Top of your list in terms of diseases or disorders we now understand so much better?
At the very, very top of my list is cancer. I think the Genome Project, knowing the sequence of the human genome, has been just transformative to the field of cancer. We understand it in a much deeper way. We understand the ways in which genes interact with the environment. And the wonderful thing is it is producing new kinds of therapies that are so much more specific and nuanced than the ones that were in prior use. So it is changing the face of cancer. The other thing I would mention is we are at the dawn of the field of gene therapy. There are in late-stage clinical trials now some simply extraordinary therapies to treat simple genetic diseases, the kinds that Aravinda has studied for many, many years. And I think in the next 10 years, we're going to see transformations in the way in which some of these very, very simple genetic diseases like sickle cell anemia, to give you a very good example that's current, is potentially on the brink of being cured. Who would have thought?
Aravinda, can you amplify on that? Because the diseases that one generation passes down to the next and the next and the next within a family tree, how much has understanding the human genome helped us make progress against those?
Well, there are two kinds. If you look at the rare or sometimes not so rare diseases, just look at what happened in your own city in 1989. The gene for cystic fibrosis was identified, by a group of researchers at Sick Kids, together with other researchers in the U.S. And you know, that disorder, which was completely not understood except by inheritance, that it was passed, and we knew the rules by which it was passed from parents to offspring, went from 1989. Yes, it did take 20 years. It took somewhat more than 20 years, but they're very effective therapies for many forms of cystic fibrosis. I think for many others, just as Shirley mentioned, cancer, I think genetics has clearly been a success story. That genetic understanding has helped us both diagnose cancer as well as monitor cancer as when it metastasizes, as well as think of therapies. I personally believe that there'll be many such areas. I think cardiovascular disease is the other big one. It's one of the leading causes of death the world over, from defects in cholesterol to defects in various kinds of heart rhythm disorders. And I think we are just at the cusp of trying to identify, but not only identify, also intervene in a much more logical and a much more planned way. So I think if, as humans, we can mitigate and reduce the effects of both cancer and heart disease, we would have gone a long, long way.
And David, what about psychiatric disorders? How much better are we at understanding those thanks to the work that you and others have done?
We haven't made nearly the progress in psychiatric disorders as we have in cancer and the other areas that Shirley beautifully outlined and Aravinda elaborated on. And why that's the case is part that it's very difficult to get a precise phenotype in some of these psychiatric disorders. But I think part of it is also the structure of the formation of the brain during the 9 months that the baby is in utero is in a very, very complex procedure that we have only a slight inkling of. And we really need to understand what's actually going on when you build a human brain before we could interpret the architecture on which these psychiatric diseases manifest themselves. Without that knowledge, it's quite difficult to interpret. And again, as Aravinda pointed out, there are many, many genes. So autism being one of the huge number, one of the diseases that that has accumulated a huge number of diseases, of genes that contribute. And that makes it extremely complex.
Shirley, if I've got my numbers right here, I think it was about $300 million that it cost to do all this work back in the day. Nowadays, you can get your genome sequence for less than $1,000. I wonder, though, how close we are to genuine personalized medicine, which, of course, is one of the great hopes that people have emerging from all of this research and information. Where are we on that?
I think in wealthy countries like the United States, I think we're there with personalized medicine. I think it is now very common if you are diagnosed with cancer, that the first thing that is ordered is a sequence of your genome and a sequence of the cancer and a comparison between the two. That then leads the oncologist to make very different kinds of decisions based on how that cancer is going to be treated. So in that sense, I think precision medicine is already in practice. It is not being practiced around the world, largely for technological issues as well as cost issues. But I am confident that we are heading into a time when we are going to be able to use genomic information to tailor treatments, therapies for many, many different kinds of diseases.
The announcement of the Human Genome Project's completion was hailed as, well, the end of the beginning and a launchpad for a whole new era. Now that we've had a couple of decades to look back at it, how did it change the work of the biologist? Aravinda, why don't you take a kick at that one?
I think the biggest change, and Shirley can correct me, I think many kinds, if not most kinds of biologists who were not geneticists had believed that they would occasionally look up some sequence, the sequence of their favorite protein. I think most biologists nowadays use genetic information and genomic information on a daily basis, and they don't even think about it twice. And to me, that's really been the significant change. I see this in my wife, who's a cell biologist. I see this in all of my colleagues. And I've seen this in all manner of meetings that are not genetics focused. Genomics and using genomic information has become really, in a way, ordinary. And nobody thinks about it as doing something special.
Well, David, let me follow up with one of the first things you said, and that was you told us that when you had the eureka moment, you uploaded it all to the internet and you shared all of that data with the whole world. I wonder whether you believe that commitment to open data and sharing information is as alive in 2021 as it was 20 years ago.
It took a hit when we got into human medicine, because there are lots of restrictions on that, but there's a culture of keeping your data for as long as you can, to milk it for as many publications as you can, to enhance your own status. And we really have to get beyond that. We really have to be more generous in sharing information rapidly and openly and working together on this problem. So I think we've slipped back a little bit in terms of openness since the genome was released in 2000, but we still have a strong commitment to working together, and that's the only way we will ever be able to tackle a system as complicated as the genetic architecture and how it contributes to disease. There are many, many examples. We have an 800-member consortium working on this difficult problem of psychiatric diseases, who collected information from 500,000 normal people and 200,000 people with different diseases. So we do have large collaborative efforts now, and they are starting to unravel this complicated network. But the future is in more sharing. And coming from computer science, I would like to see much more efforts that are like open source efforts in computer science, rapid publishing, everything open. This will speed things up.
Shirley, I did quote Richard Gibbs earlier, who also believes that there tends to be a 25 to 30-year lag time between discovery and true profound impact on healthcare delivery, for example. So we're getting close to that timeline right now. Do you believe we're on the precipice of something big?
I wouldn't say that there's going to be sort of a critical tipping point where suddenly there's a flourish of many new advances that affect human health. But I will say that I think Richard was correct, that turning knowledge into medicine is a long, hard journey. And I think we embarked upon that journey when we got the sequence of the genome. And we're going to be on that journey for many years to come. So I think the advances are going to come, but probably not with an avalanche, but with something that's a little better than a trickle, but not an avalanche.
Aravinda, how about you? Avalanche or trickle?
No, I think it's somewhere in between. I think what the Genome Project did, beyond sequencing the human genome, is to create this discipline of genomics and show how this was important. Just look at what we've gone through in the past year and still are in the midst of this pandemic. The pandemic, I think there have been two significant advances that genomics contributed to. One is sequencing the virus very rapidly, as you know, very early in 2020, that led to Moderna, in fact, creating the first vaccine. Sequencing was critical to it. The second has been this widespread testing of the virus in patients. We really all hope that this would have started earlier in the US, for example, in many countries. It would have been far more widespread. That speaks to questions of infrastructure. But there's absolutely no doubt the reason why we speak of a South African variant or Brazilian variant and other kinds of variants is simply because sequencing is revealing how the virus is changing and when the virus is apparently becoming more transmissible. These are things we could not have imagined in the absence of the Human Genome Project. At least I cannot imagine.
In which case, thank you all for your work. That's our time, everybody. David Hausler, Shirley Tillman, Aravinda Chakravarti. It's been so good having all of you on TVO tonight. Thanks so much for your time. The work done to map the human genome and begin to make sense of it is, in many ways, awe-inspiring. Understanding the potential and peril that flows from it is almost unfathomable. Jamie Metzel is a senior fellow at the Atlantic Council and a member of the World Health Organization's Expert Advisory Committee on Human Genome Editing. His most recent book is called Hacking Darwin: Genetic Engineering and the Future of Humanity, and he joins us now from Vail, Colorado to point the way ahead. Jamie, it's great to meet you. How you doing?
I'm doing great, Steve. Happy to be here.
Excellent. You write in the book that after nearly 4 billion years of evolution by one set of rules, our species is about to begin evolving by another. What's the new set of rules you're referring to there?
The new set of rules are humans driving our evolutionary process forward. For billions of years, we've been guided and driven by random mutation and natural selection, which are the foundations of Darwinian evolution. And now our one species among the billions that have ever lived suddenly has the ability to read, write, and ultimately hack the genetic code of life. And that gives us awe-inspiring, almost godlike capabilities. And the question for us is, can we use them wisely?
Well, humor me for a second here. If we go down the road half a century, what will people then be saying about the time in which we're living right now, apropos of what you just said?
I think they'll be saying that this moment was a quantum leap moment for the technologies of the genetics and biotech revolutions, just like World War II significantly pushed forward electronics and space travel. We're seeing that with these almost miraculous mRNA vaccines. What those vaccines are doing is inserting genetic code into our bodies, instructing our cells to produce something that wasn't already there, in this case, the spike protein of the SARS-CoV-2 virus, and then to respond by creating an immunological response to what we have created. That's just one example of how we, our one species, is hijacking the tools of biology, and that's going to fundamentally transform not just our healthcare, but so many aspects of our lives, from the way that we make and consume food and agriculture, to how we use energy, to materials, and ultimately will transform our evolutionary trajectory as a species.
Let me take us off the path just for a moment here, because I want to find out more about you. You're a guy who came up during the Bill Clinton administration. You were on the national security team there. Terrorism was, the issue of the day. And suddenly here you are, a guy interested in our genetic future. Somewhere along the way, you came to a fork in the road. What happened?
Yeah. So when I was working in the Clinton, thanks, Steve. When I was working in the Clinton administration, terrorism was something that my little office was focused on, but the U.S. government wasn't in a meaningful way focused on terrorism. My boss then and good friend now is someone named Richard Clarke, and he was the Cassandra of the terrorist threat. He was telling everyone who would listen that there was this big and fundamental threat that was coming and that we had to respond. And when 9/11 arrived, Dick, unfortunately, was the person who had sounded the warning, but the warning hadn't been heard. And Dick always used to say that if everyone in Washington or the world is focusing on one thing, your value add isn't focusing on that thing. It's trying to say, well, what are the big issues that aren't being addressed, that aren't being explored? And so for me, nearly 25 years ago, as I looked around the world, I came to feel very intensely that the tools of the genetics and biotech revolutions were going to change everything. So I started educating myself when I was ready, started doing a lot of writing, started testifying before Congress. Then I was doing speaking, and I wasn't getting through. So I wrote science fiction novels trying to tell this story in ways that people could more readily absorb. And when I was on my book tours for those sci-fi novels and I explained the science to people and they suddenly got what was at stake, I could see their eyes going wide and they recognized that something big and fundamental was happening. And that was when I realized that I needed to write what I hope is the book on our genetic past, present, and future. And it's been really an incredible journey. I'm now, as you said in the intro, a member of the WHO International Expert Advisory Committee on Human Genome Editing. And what we're trying to figure out is, how do we govern what could be the most powerful technology in all of human history in a way that optimizes the benefits and minimizes any potential harm? But as always, there's a race between, in this case, an exponentially advancing technology and governance and regulatory infrastructures that maybe even by definition only inch forward glacially. And because the speed of scientific advancement is increasing for all sorts of reasons, that mismatch becomes all the greater. And just to put some numbers on that, last year in 2020, the Nobel Prize in Chemistry went to Jennifer Doudna and Emmanuel Charpentier, who came out with a brilliant paper in 2012 describing the CRISPR-Cas9 genome editing tool, but it was really just a basic science paper. 6 years later, in 2018, the world's first CRISPR babies were born in China. This science is racing forward, and we're not ready for it. So I don't know whether I'm more or less successful, but certainly we are not successful enough at laying a foundation to make sure that we can use these very powerful technologies with maximal application of values and ethics.
Well, you mentioned babies, and I want to stick with children and parenting there if we can here. It is becoming, of course, much cheaper to sequence the human genome, and parents can, if they want to, some parents anyway, they can learn a lot more about their children's genetic makeup. Do you think this has the potential to change parenting going forward?
I think it has the potential to change parenting, and I think it will change parenting. But we also need to recognize that parenting is always changing. So when we have information about our future children, just basic information that we learn from our children. For example, oh, my kid is really good at sports or music or whatever. We internalize that information and we try to give those children more opportunities. So if we have that information and we understand that our future children have a genetic propensity to being really good at one thing or another, will that increase the likelihood that we expose them to that thing, whether abstract math or something else? I think it absolutely will, and maybe even in some cases it should. But what we're talking about is something even deeper than that, because this is not just going to change the way we make babies, but the nature of the babies that we make. So when does parenting begin? Well, maybe in the near future, or even now, parenting begins when you decide how you're going to have your offspring, whether through sex or through using the newer tools of in vitro fertilization, maybe embryo screening. And that embryo screening of pre-implanted embryos is going to be super empowered by our increasing knowledge of the genome. And although the first applications of genome editing to pre-implanted embryos in China was highly unethical and should be condemned, I think it's extremely likely, almost certain, that we will be using the tools of genome editing in select limited cases on pre-implanted human embryos at some point in the future.
Wow, I got to follow up on that because that is absolutely fascinating. So you see a future where more and more parents-to-be, are going to opt for IVF, even though they don't have to, because they want to genetically re-engineer the children that they're going to have? Do I hear that right?
Not necessarily because they want to genetically re-engineer their future children, but at least at first because they want to reduce the risk inherent in being a human being. And that's our entire healthcare systems are based on reducing that risk. 1 to 2% of all children are born with terrible and in many cases deadly genetic abnormalities. If we can prevent that or fix that before these children are born, so that rather than living 3-year painful lives before terrible deaths, these children can live 90, 100 years, I think people will be very comfortable with that. And we need to recognize that we, in the way that we're living now, we are not living in some perfect state of nature. Our lives are very, very different from our ancestors because we are comfortable using technology. Agriculture is a technology. Medicine is a technology. Getting vaccinated so you don't die from smallpox is a technology. This is also a technology which, if used wisely, can enhance our humanity, and if used poorly, can detract from it.
Well, let me pursue that, because I suspect there's not that much debate about using this ability in order to prevent a child from having a horrendous life filled with health travails, etc. Not much discussion there.
But there is.
Well,
in fact, there is. There's a whole community of people who are saying that the genome is sacred, and any engineering, any manipulation of the genome, even to make a heritable single gene mutation to eliminate a terrible, deadly disorder, there are a lot of people who feel that is crossing a line and quote unquote playing God. I don't think so, but that's why I think we need to do that carefully. But sorry to interrupt you.
No, fair point. But I guess where I was going with this was that's one set of ethical considerations. Another set of ethical considerations entirely, where I think you may get onto a more slippery slope, is how about messing around to turn an introvert into an extrovert? How about trying to mess around in order to have somebody be more empathetic or more intelligent or more athletic or something like that? Where are you on the ethics of doing that?
So all of these things are extremely thorny ethical questions. And first we need to, when you use the word messing around, well, we need to say, well, what do we mean by messing around? So one would be messing around we have everyone's genomes are sequenced and we get a sense of who is likely to be a world-class sprinter. That doesn't mean they're going to be, but who has the potential to be. And when we try to encourage them to step in that direction, that's the Plato's Republic model. Another option, which I've written about in my editorial not too long ago in the New York Times, where I was imagining a visit to the fertility clinic in 2050, would be in the process of IVF, when screening some number of embryos, whether it's 10 or 15 in our world today, or let's call it 10,000 if we use stem cells in the future to expand the number of eggs, and then choosing, well, you have to choose one of these embryos to implant in the mother. And then maybe people would want to screen for health, but maybe people would want to screen in the future for certain genetically determined or largely genetically determined outcomes. And then the third option of messing around could be using the tools of genome editing to increase the likelihood of one particular outcome or another. But because our genetic traits, most of our traits, frankly, are so complex, many different genes, and then interacting with environmental factors, it's not going to be that having a baby is like going to the Build-A-Bear showroom in your local mall where you just pick attributes, at least for many, many hundreds, if not thousands of years. But I do think that we will increasingly use the tools of embryo selection and over time limited genome editing. But these are very, very big issues that come with lots of critically important ethical issues that we need to wrestle with.
Well, to that end, I mean, if you're a Star Trek fan, there's a bit of Khan, Noonien Singh happening here. Do you see a future that is more concerning in this area or more exciting in this area?
All in all, I am an optimist. Our species has been using all of our technologies to live our lives, to do things that at least we've come to believe are better. Would we have been better off without harnessing fire, without agriculture, without medicine, without air travel, without the internet, without all of these different things? I'm sure there are some people who are saying yes. These are technologies like every other technologies that can be used well or can be abused. When we imagine what it looks like when they're used well, we live longer, healthier, more robust lives. We enhance our humanity. We increase our capacity to have more healthy time to love each other, to write poetry, to innovate, to do all the things that make humans such a special species. If we abuse these technologies, we could use them to crash ecosystems, to diminish our humanity, and to do lots of terrible, terrible things. And the difference between those two outcomes is us. And that's why I'm spending so much of my life's energy trying to push forward an inclusive global dialogue on how we should best use these technologies in ways that enhance the benefits and minimize any potential harms.
I appreciate the religious argument that says we ought not to be playing God and we should not involve ourselves in any genetic changes at all. Period, full stop. That's a religious argument. I understand it. What is the scientific argument that says it is unethical for us to, if I can use my scientific term again, mess around here, to, for example, get rid of Alzheimer's forever, or sickle cell anemia forever, or cystic fibrosis forever? What's the argument there?
So first, let me talk about the religious argument. I think it's very legitimate. I mean, these are very powerful technologies. No one group of us can railroad anybody else. As a matter of fact, I was very honored just before the pandemic to have been honored to speak at the Vatican on exactly these issues. And I was a little surprised, but very happy when they commissioned me to write a paper on the Catholic Church and human genetic engineering, which they've published and people can find on my jamiemetzl.com website and blog. So there are strong religious arguments that I think we should consider. At the same time, there are some people, and I meet them from time to time, who believe that terrible human suffering is fate, or some people even believe it's a contribution to the rest of our humanity. I believe that the history of our species has been fighting against these kinds of terrible things. When somebody has a child who's dying of a terrible genetic disorder, I think those parents are saying, well, what can we do to fix that? If you gave them a tool to prevent that, most parents would want that. And so it's not crazy for us to say we're going to use the tools that we have to fight and prevent terrible and deadly genetic disorders. Everyone should ask the question, if it was your spouse, your parents, your children, would you want that? And I think the answer is yes. And so if this gets framed as an everything or nothing debate, we're all losers. What we need to do is find a sensible, wise middle path that can bring us all together and allow us to use these technologies in the best available ways that follow our most cherished ethical traditions.
Well, Jamie, let me make things just even a little bit more complicated. We're coming to you from Canada today. Canada is a single-payer healthcare system. We think equity is one of the absolute sine qua nons of our healthcare system. This notion of equity, what if we want to introduce the kinds of possibilities going forward that you have described to us tonight? But it's expensive, and public healthcare systems or publicly funded healthcare systems just won't cover it. We get into a circumstance where the rich will be able to afford to do this, the less well-off will not. What are the problems around that?
It's a really great and important question. And first, let me say that I, for one, am jealous of the single-payer national healthcare system that you guys have in Canada. The United States is great for super specialized, high-cost interventions, but in terms of creating a public health infrastructure that helps everybody, Canada is a great model. The reason why we're getting so much great information about the pandemic from Israel is that they have a model, a national healthcare system that makes sense. And so issues of equity are absolutely essential. Equity and diversity are really the two most essential ethical questions that we need to consider. But those are choices that we are making. We are making decisions every day about how to allocate our resources and who gets what. And so I think we need to make different kinds of decisions, certainly than we make here in the United States. In Israel, for example, assisted reproduction is largely covered in national health plans. That doesn't mean that every application that anyone can imagine is covered, but some of the most important ones are. And so if we want to live in a future where we have equitable access to healthcare, including these tools of assisted reproduction, we need to start building that future now. And that's why certainly I believe the United States has a lot to learn from countries like Canada and Israel. And at the same time, there's a reason why a lot of this innovation is happening in the United States. And part of that is that we're flooding so much money into the healthcare system. But you're absolutely right. These equity issues alongside the diversity issues are fundamental to making sure that this technology can be applied in a way that enhances our humanity and doesn't divide us or weaken us.
At some point down the road, somebody is going to have to decide how this is all going to work. Who's that somebody?
So I'm spending a lot of my time trying to address that question. As you mentioned in the intro, I'm a member of the World Health Organization International Advisory Committee on Human Genome Editing. And what we're trying to do is at least recommend what a governance framework for these technologies might look like. And it's really complicated because in our world today, it's not just governments, it's not just the UN. Governance happens at so many different levels. And so as I see it, we need to have a top-down, bottom-up approach. From top-down governments, we need to make sure that every government has a wise regulatory structure in its own country that represents both international practices and the unique culture and history of that country. We need to make sure that we need new treaties on an international level. But perhaps most significantly, we need what I call a species-wide dialogue on the future of human genetic engineering that can begin a process of developing norms of behavior that can underpin all of those other things. And so that's why there's a race. There's a race between this exponentially growing technology and our ability to establish governance systems to use it wisely.
We know what mutually assured destruction was back in the Cold War. I wonder whether, given the scenario you've just described, if we have different countries pursuing different approaches, do we have a mutually assured, genetically altered future coming our way sometime in the future?
Well, I write a lot about that in Hacking Darwin. I call it the arms race of the human race. And if we aren't careful, we're going to have that kind of arm race situation. I write about this in the book, but it's not coincidental that the world's first 3 CRISPR babies were born in China, and the Chinese biophysicists was an extreme nationalist in everything that he was writing. He talked about bringing glory to China. So these technologies exist within the cultural context of us, of how we have organized ourselves. And if we aren't careful, if we don't try to find the right balance between local, national, and global governance, ethics, and norms, and we will be in that kind of situation. And we will be, if this technology works as well as some people think, in a situation where one country is intervening, and whether it's through embryo selection or genome editing or something else, in ways that are considered, rightly or wrongly, to give that country an advantage. And then other countries have to decide, how do they respond? Did they do nothing? Do they try to stop the one country, or do they try to match it? And that is a plausible scenario for the future, which is why we have an incredible urgency to try to do everything we can now.
Name of the book is Hacking Darwin. Absolutely fascinating stuff. And it has brought Jamie Metzl to our virtual studio anyway, from Vail, Colorado. Jamie, it's great to talk to you. You stay safe and be well, okay?
All right. Thanks so much, Steve.
And that is the agenda for Monday, March the 1st, 2021. 50 years ago today, incidentally, Bill Davis sworn in as Ontario's 18th Premier. I knew you'd want to know. Moving on, the vaccination picture changed last week when Health Canada approved a third vaccine for use here. Tomorrow, we'll follow the timeline of what it took to start getting Canadians inoculated. I'm Steve Paikin. Thanks for watching TVO, for joining us online at tvo.org, and we'll see you again tomorrow.
The Agenda with Steve Paikin is brought to you by the Chartered Professional Accountants of Ontario. CPA Ontario is a regulator, an educator, a thought leader, and an advocate. We protect the public. We advance our profession. We guide our CPAs. We are CPA Ontario. And by viewers like you. Thank you.